Welcome to my first screencast. We're going to begin with enzymes, which is going to be the subject of our third and final SAC for Unit 3. The most important thing to remember about enzymes is that they are proteins. And like all proteins, they have 3 or 4 different levels of structure. The first level of structure, as we see here, is the primary structure. This is basically just the amino acid sequence. They've been strung together using the usual method of a condensation reaction, where the different amino acids are joined each time releasing a molecule of water. The second structure is the secondary structure, and this will either be seen as a beta pleated sheet, seen here on the left, or as an alpha helix, seen here on the right. There is a third structure, which is a random coil. That is really just a sequence, the amino acid sequence, without any formal structure to it. The third structure that we'll see here is the tertiary structure. That relates to the way that all of the random coils, the beta pleated sheets, and alpha helices have been all arranged. This is the most important part of the structure, as far as we're concerned when we're dealing with enzymes. If the tertiary structure changes, then the whole properties of the protein itself changes too. Occasionally we do see enzymes that go into a quaternary structure, which we see down here at the bottom, and the quaternary structure is really just multiple versions of the tertiary structure stuck together. It doesn't always happen in a protein, but when it does, it's just as important as the tertiary structure. Proteins are also organic molecules, so they all contain certain varying amounts of carbon and hydrogen. They will also contain nitrogen. Nitrogen is vitally important in the amine group, which is what gives amino acids their name. There can also be a range of other atoms involved, but they are generally in much smaller quantities and are specific to the particular proteins involved. The tertiary shape of a protein is very, very specific, especially with enzymes, because they have an active site which needs to be a particular shape. If for any reason this shape is damaged by various factors, which we'll go into in a moment, then the protein will denature, or at the very least change its properties so that it no longer performs the job that it used to. This slide shows just how complex a protein can be. And in this case it's the enzyme glyceraldehyde 3-phosphate dehydrogenase, which is involved in glycolysis. You can see that it has NAD molecules attached to it in specific spots, and those are cofactors, but we'll come back to that later on. We can see that it has a quaternary shape because we have 4 separate tertiary structures stuck together. Alright, now down to the nitty-gritty. What do enzymes actually do? Enzymes are often considered to make reactions go faster. That's more of a byproduct of what they do. What they really do is lower the activation energy, and this has the effect of making the reactions go faster given the amount of energy that's already available in the environment. We can see on this diagram that the energy required without the enzyme is fairly high. You can imagine it almost as being like a hurdle, and the reaction is on the other side. the effect that the enzyme has is to lower that hurdle and make it much easier for the reaction to progress. You could consider it to be like starting a fire, either by rubbing two sticks together, which would take a great deal of energy, or striking a match. The enzyme is essentially working like the match and enables the reaction to happen much easier and requiring less energy. Right, how do they do it? We can see from this little picture here that the enzyme, which is the purple structure, has got a very particular shape. Let's write enzyme in here. The enzyme fits very nicely with the substrate, and when the substrate fits into the enzyme, a particular part of the substrate is worked upon by various cofactors usually, and it causes a bond there to break. In this particular case, sometimes it's possible that energy can be provided and a bond will form, so this entire thing can go backwards. We mustn't always assume that an enzyme catalyzed reaction will be breaking things. Very often it will be making things as well. And we can see at both ends of the reaction that the enzyme, this end and at that end, are the same. In both cases, whether you're building something or breaking it down, the enzyme is not destroyed, usually. Eventually they do wear out, but generally with each and every step, the enzyme is able to go back again and do the whole thing all over again. So it doesn't get broken down, but the substrate either gets broken down or reformed into something bigger. Okay, what affects enzyme activity? I've included this picture here because it's fairly typical of a protein. We can see that we have our alpha helix, like there, and another one here. And we even have some beta pleated sheets in the middle. There's two of them side by side running through the middle there. And we have our random coils in various places joining everything together. What this means is that anything that affects the tertiary shape of this protein, that picture that you see there, anything causes it to alter in shape will affect the way the enzyme works. I've got 5 separate factors listed here. We have pH, temperature, enzyme concentration, substrate concentration, and some inhibitors. Every enzyme has an optimal pH. We can see in both cases here that they have a peak to their rate of reaction, and that peak is where their optimum is. We can see with the first one on the green line, this is its optimal pH is around about 2, and the red line, its optimal pH is around about just under 7. We can say with reasonable certainty that the green line is it's probably going to be an enzyme like pepsin. We can tell that because the optimal pH is so very low. You'll only get a pH like that in our stomachs, and so the enzymes that work in our stomachs have evolved specifically to work at those very low pHs. The rest of our body is generally a pH of around about 7, sometimes slightly higher, sometimes slightly lower, depending on where you're looking. So we can say with reasonable certainty that the red line represents pretty much most of the other enzymes in our bodies. We can also look, when we're looking at the red line, we can see that if we go beyond 7, then it starts to become very alkaline and the rate of reaction drops off quite sharply. That's because we're going outside of the optimal range for that particular enzyme. On the face of it, the graph for the effect of temperature on enzyme activity looks very much like the graphs that we saw for pH, but with one important difference. At the lower end, when temperatures become very cold, the enzyme activity tails off. It goes down to 0, but the enzymes are not broken down. At the other end, we can see that let's go over to blue colour, we can see that the temperature, the activity drops down to 0 very suddenly. At this point, the high temperatures have denatured the protein completely and it is dead. Essentially, it's like boiling the egg white, making it go white. We have changed the tertiary shape of that protein to the extent that it no longer is able to function. At the other end of the scale, when we are dealing with very cold temperatures, we still have that tertiary shape. It hasn't denatured at all. The rate of reaction has dropped significantly because of the way that the molecules have now stopped moving quite so much. In both cases, we can see, with both pH and temperature, we can see that we have an optimal. That is a peak to our enzyme activity. This is for roughly between, let's say, about 37 and about 45 degrees centigrade. So for that area there, the enzymes in our body are working at their peak. It doesn't necessarily mean that it's comfortable or that it's optimal necessarily for the rest of our bodies, but for this particular enzyme it's working at its best at those temperatures. This contrasts with the graph of pH and activity, in that when pH becomes either very low or very high, so either become very acidic or very alkaline, you can often get denaturation of the proteins at both ends of the scale, but we don't usually see that with temperature. The graphs for pH and temperature are completely different to the one for substrate concentration. In this graph we can see that the rate of reaction increases quite nicely, and then it reaches a point at which it all stops and it levels off. That doesn't necessarily mean that the reaction has stopped happening, but it means that it's no longer going any faster. As we're increasing the amount of substrate that we have, if we consider the substrate to say be a sugar solution, the more sugar we add to that solution, we're not necessarily getting any more product being formed by the enzyme in the solution. This is for the simple fact that there are only so many enzyme molecules in that solution, and once they're all working as fast as they can, they literally can't go any faster, and so that's when we see a maximum rate for the rate of reaction in this case. There will be other limiting factors that come into play once we start to then increase the amount of enzyme, but we'll look at that in the next slide. Alright, this is the slide for enzyme activity and with regards to the enzyme concentration. I'm not particularly happy with this graph, I have to admit. I was in two minds whether or not to include it, because what this graph is assuming is that we have a virtually unlimited amount of substrate. So the more enzyme that we have, it's increasing, the rate of reaction is also increasing. But at some point we are going to have another factor start to limit it. That factor could be a build-up of product which may inhibit the enzyme. It could be temperature, it could be pH. Things like that can end up changing when the rate of reaction gets very high. So eventually we can assume that this graph here will start to level off as some other factor starts to come into play. And the most obvious factor in my mind would be the substrate concentration. So ultimately the graphs for substrate concentration and enzyme concentration will end up looking very similar, but the slopes and the point at which they level off will be different. Alright, so what can inhibit enzyme activity? There are various things in the natural world and in the man-made world that will do that. Many things like the brown husks on these almonds contain chemicals which inhibit the enzymes that allow the almond to germinate. The only way that these inhibitors are removed is with large amounts of water and very often things like bacterial action. So really these almonds need to be in the ground for some time for those inhibitors to be leached away or broken down, and then the almond itself will start to grow. The other sort of inhibitors that we find, and we will get into these more when we start dealing with the nervous system, would be various man-made chemicals. The most obvious ones would be insecticides. Generally there are two different sorts of inhibitors. The first one is a competitive inhibitor. The most important thing to remember about a competitive inhibitor is that it competes with the substrate here, a little green molecule, for the active site. And the active site is that little V-shaped notch in the enzyme itself. We're assuming here that this particular enzyme uses that key and lock mechanism to bind with its substrate to form an enzyme substrate complex, and once that happens then the enzyme will often distort slightly and the substrate will be worked upon in one way or another. Remember, we're not always going to be breaking things down as we see in this picture. We can often be making things or just altering them in some way. Now what the inhibitor does is it will come along and it will have a site very much like the substrate that should be binding, and that will fit neatly into the active site. Unfortunately for the enzyme, when it does this, it then doesn't behave in the way that the enzyme substrate would. In this case we can see that it doesn't actually break. Unlike the enzyme substrate that we see above, it retains its shape and it stays locked into the enzyme. This essentially ties up the enzyme until it degrades and is broken down, so that enzyme is no longer able to act at all. This can have rather serious consequences for the organism which is relying on this particular enzyme. If this enzyme was to be like that large enzyme that we saw in slide 2, where it's part of glycolysis, if that particular enzyme was being seriously inhibited, then glycolysis would at least slow down dramatically or stop, and the entire chain of respiration that follows after that would break down, and basically the animal would die. The same thing would happen in any of the various steps that you see in photosynthesis in plants. The other sort of inhibitor is a non-competitive inhibitor. And the thing about these ones is that they do not bind with the active site. If we look at our picture here, we have a little star-shaped inhibitor, and what that does is it doesn't bind in the active site, which I shall mark in red here, which our nice neat little substrate would fit straight into very nicely. What it does instead is it goes round to the other side and binds with another part of the protein. If we think back to that big complex picture in slide 2, there is the potential for there to be many sites in which other things can bind on a very complex protein molecule. It just so happens that in enzymes there is a nice little spot for the substrate to fit into, but there are always other places that inhibitors can go into instead. In this case, once the non-competitive inhibitor has affected our enzyme, we can see here, if we zoom in slightly, we can see that it no longer has its nice neat hexagonal shape. We have this extra bulge in it instead, and that's been caused by the electromagnetic field of our inhibitor here, and that has led to a change in the tertiary shape. Now, it's not always the case that this is irreversible, but if it is, then it means exactly in the same way that the competitive inhibitor has done, it now ties up that enzyme forever and it can no longer do its job. The last things we need to deal with when looking at enzymes is the other bits and pieces that work along with them. There are two different sorts. Coenzymes are organic molecules. That means that they also contain carbon and hydrogen in some form, along with various other things. Coenzymes and their inorganic counterparts, cofactors, fit into the active site. They alter or improve the shape of the active site, as we can see in this picture. The active site is no longer like so. It is now more like that. And what this does is that it will change what substrates can be acted upon by that enzyme. Now, very often, cofactors can be things that supply energy for the reaction. And the two main ones that we have been dealing with already are ATP and NADH. Both of these are sources of energy in metabolic reactions. What they do is they will fit into the active site, and they will supply energy to either break or form bonds when the reaction is happening. The main coenzymes that you will see around are very often vitamins, and they simply improve the way that the enzymes work, but they're not quite as important, certainly in terms of the VCE, as cofactors are. Now, there are two other terms here which we see down the bottom, apoenzyme and holoenzyme. Let's just do what we need to with those and wipe them out. We don't need to know that. That's more terminology than we really need. And the term that they use there, catalytic site, just getting my pen right. We don't call it a catalytic site, do we? We call it an active site. Well, this brings us to the end of my first screencast. If you have any questions or comments, please post them to the Edmodo page. I have undernard probably a little bit more than I would have liked to in this, and I have to admit Douchy does make it look very easy when he does his podcasts. I would strongly recommend that you listen to his podcasts. He's done one quite recently on enzymes. You can find them either on iTunes, but if you Google them, there are a couple of websites out there that host them for him.